Okay, so we know that there are cells that continuously divide and they produce more of themselves. And one such way to do that is also known as mitosis. Now, mitosis is a part of it is also known as the phase in which a cell divides is known as the M phase, which is quite different than all of the other phases of the cell cycle. Mitosis is actually characterized by the way in which the chromosomes by the way in which the DNA in this stage is noticed is found. So the DNA in the stage of the M phase of a cell cycle is much more compact. So it's much more compact as compared to the other phases. And the first phase of the M phase is basically the formation of the mitotic chromosomes or more compact chromosomes. If you look at the other phases, if you look at this cell in the interphase here, you can see that most of the DNA in the cell in it, which is in the interphase phase of the cell cycle, the the DNA is quite thread-like and dispersed all across the cell. But as soon as the cell progresses to the M phase, you can see that the DNA is much more compacted and much more neatly organized. Now the question here is how and why does this happen? This can be explained by a molecular entity known as condensins. Condensins are a result of CDK activation. CDK are just some checkpoints that a cell need to pass when it passes need to activate when it passes from one phase of the cell to another. So, for example, if there's a passage between the M to the G1, there's an activation of CDK that leads to it recalls different proteins to carry out the processes that happen in the next stage. And one such protein is the condensin protein. Now, the condensin protein does not really like to work alone. It works in close association with two other molecules. One is the topoisomerase 2, and the other is the usage of ATP. So, topoisomerase 2 and ATP essentially activate the condensin, and the condensin then, it neatly packs this thread-like DNA in the interface, and sort of puts it into a neat X-like structure in the cell in the M phase. Now let us again take a look at the typical cell at the interface of the cell cycle. Now, in the interface, you can notice that most of the DNA that is present inside the cell is in the form of a thread-like structure. It's not really organized. So we know for the fact that there are certain molecules that help in the compaction of the DNA, and these are known as condensins. But then there's another molecule, as you can see, another protein, these blue small dots that are also associated in the compaction and the maintenance of the integrity of the cell in the next phase that is also known as the M phase or the mitotic phase, especially in the prophase. As you can see that the protein that is noted by blue here is also present in the M phase. So it moves from this phase and aids in especially cohesins. These proteins are known as cohesins. Cohesins, they help in the compaction and also they act like clips. So basically there's the basement, so there's the skeleton of the cell and that's the chromosome, and they sort of help in order to clip this chromosome right onto the scaffold or the skeleton. And then they also sort of bundle it up together, and their structure is similar to the condensins, but they have slightly different functions. Now, if you look at the typical cell cycle, you can notice that most of the cohesins that are found are concentrated in the G2 phase. And so G2 phase is when the chromosome is in a thread-like structure, and then they go all the way into early M phase. And what is early M phase? Early M phase is essentially prophase. Now, we don't really need cohesins to go all the way to till the end of the cycle. So we need the removal of cohesins. Because their only job is to, sort of arrange the chromosomes and then, get it ready for the mitotic or the metaphase and we need to get rid of it before the progression from prophase to metaphase. If you look at a typical, sorry, if you look at the typical prophase chromosome in a typical prophase of the M phase, you can see that it's an X-like structure, and all of these blue structures that are present right here are the clip-like cohesins that associate that hold the entire chromosome onto the cytoskeleton, and the removal of these structure is mediated by two kinases. So we have polo-like kinases and aurora B kinases. Kinases essentially phosphorylate these molecules and remove them from the chromosome. And then we have the VAPL kinase that essentially does the leftover, job, or just takes out the leftover particles. So after the job is done, the chromosome looks something like this. But as you can see that we still have a bit of kind of cohesins that are remaining in the centromere of the chromosome. But why is that so? So the a lot of phosphatases that are present in this region that do not allow the kinase to phosphorylate the cohesins that are found in this region. And this is essential. This is quite essential for the chromosome um, because we need the sister chromatids to be associated together and inside, until the anaphase at least, so that there won't be any unnecessary separation until the late metaphase or anaphase. We know for the fact that chromosomes separate during anaphase, and until then we need the conjunction of two sister chromatids, two homologous sister chromatids together to form the chromosome.